Thank you very much. As already mentioned, I'm a little bit of an imposter because I'm a microbiologist. In particular, I'm a synthetic biologist. It's kind of my thing, and that's what I do. So I'm hoping, unless I've done things hilariously wrong, which might be true, a lot of the Agile concepts in here you'll already know. So that'll be fairly familiar to you. You can take a breath. That's good. The thing that will be new is the biology aspect, some of the weird nuances of trying to apply Agile to microbiology, trying to apply it to synthetic biology, and how that kind of works. If what I'm talking about is actually really boring and you're really used to it, there should be some QR codes as we go through, which will just be random links on the internet, if your Wi-Fi is even working, that is. That will just be interesting tidbits and various side points. So if it's really boring, at least you can feign interest by looking at those. It's kind of the hope. So a little bit of background to me. I did start out my education in a town called Mansfield, which is in Nottingham. For those of you not familiar with it, it can be charitably described as challenging. I was an average student at best, which meant by some stroke of luck, I managed to get into Lancaster University, where I studied biomedical science. From there, I got involved with the Microbiology Society, and I got fascinated with bacteria and tiny things and how they operated. In particular, things like the flagellum, which is a motor system that allows bacteria to swim, and how it's about 30% more efficient than a petrochemical engine, even at that tiny scale. Things like quorum sensing that I would later on to go on to study in my PhD, which is the ability for microorganisms to communicate and have little conversations in nature. This got me involved with the Synthetic Biology Research Centre in Nottingham, where I studied to do my PhD. And I spent some years there, which I'm going to talk a little bit about academia in this talk. My funding body allowed me to do an internship where I met some guys at a conference that were using a robot to pour vodka shots. And that was Singer Instruments, who I would later be hired by, and for some reason they took a risk on me. I was their scientific advisor for a while, which is kind of like a sales role, and they gave me the opportunity to lead a small team of scientists, which I still do to this day. And a lot of the information from this talk will come from that experience. I set up their quality team because I realized they needed one, and then later on was joined by their service and support team, which gives me the strange title of Service and Support Quality and Research Manager. And when you work in a small to medium enterprise, if you haven't got 3 jobs, then you're not doing it right. So synthetic biology, what the hell is that and what I'm going to be talking about? Hopefully a lot of the people in this room will understand what I'm talking about on the left here. You can see I'm an academic. There's a lot of data-heavy slides, so apologies. This is kind of the application, if you like, of Moore's law, the fact that transistors were getting cheaper and cheaper and cheaper from the kind of late 1960s and 70s through to the 2000s. And obviously, the internet was born. Companies kind of moved to digital, and there was this huge revolution in how we see the world. A lot of commentators right now are looking at similar data, and this is old data now, it's way cheaper than this. But actually with sequencing DNA, the building blocks that make us who we are, is becoming cheaper and cheaper and cheaper to understand. On top of that, DNA synthesis, the ability just to print DNA from nothing, not relying on an organism to make it, is also becoming cheaper. You can now buy devices that sit on your desk and print DNA. And the argument then is, if we kind of saw a digital revolution, perhaps we're moving towards a synthetic biology revolution. And you can see this in companies like Spiber, which is a Japanese company that take the genes to create spider silk and brew silk, and they're using it to make jackets, clean up the garment manufacturing industry. You can see it in companies like the Impossible Burger that take the heme group from a certain plant, and they use it to brew blood that they can then put into vegetarian burgers that bleed, and you can have rare. That might not be your cup of tea, but it sells particularly well. So there's huge amounts of interest in this field. So we're going to split this talk into about 4 sections, the old school academic in me coming out. First off, we're going to talk about why biology is hard to plan, a bit of history and some tips or lack thereof on project management from academia. In section 2, I'm going to talk about learning to walk before we can sprint. That's probably not new to you guys, but it might be a different application on sprinting and how that works. Section 3, I'm actually going to demo a sprint. I'm going to give a demo that was from my team just to give you an idea of the kind of things that we work on. And finally, getting back into the garage, we started achieving some objectives, but we were learning something from the past. And I'm going to leave a really big hanging open question in my talk because it wouldn't be Agile if we figured it all out, I guess. So there's going to be a big open discussion at the end. I will talk about what Singer do, so it'll come in later in the talk, so don't worry. But section 1 is problems with biology, which is kind of a funny thing to say. Normally something you'd mention to your GP, I guess. So there's a brief note on PhDs. If you are interested in doing a PhD, you're probably mad, but that's fine, we all are. But there's a great book called How to Get a PhD that I would wholeheartedly recommend. But as a bit of a crash course, it's a piece of research adding into a particular field. They take kind of between 3 or 4 years if you're within the UK. Every country's different, just to make it more maddeningly complex. But year 1, you try and figure out what the hell it is you're doing, because you've got no idea. It's called research for a reason, right? Year 2, you kind of know what you're supposed to do, but everything you try fails. Everything. We call it the second year depression or the second year slump. It's very, very common. So everything fails in your second year. Right in your third year, something might just work, if you're lucky. One of the things you're trying works, and you're fascinated by it. Like, I've done a thing, and I've changed the world, and it's going to be amazing. However, around about 3.5 years, or if you're lucky enough to be on a 4-year PhD, no time to investigate it, you've got to write a book. You've never written a book before in your life, and you have no skills in journalism or writing, but you have to write this book. And when it's finished, you get grilled for a minimum of 4 hours by 2 other academics, who then have to agree that it's good enough. And if you get academics in a room and try and get them to agree on something, you're going to be there for a very long time. So that's kind of a crash course in how PhDs work. So academia and project management don't traditionally work well together, as this cartoon in the top left kind of demonstrates. There's a very different interpretation of how deadlines work, and people are pretty lax about them in general. They're paid to be researchers and find things interesting. They're not necessarily meant to be project managers. And this here is a genuine ordering system that I've seen in an academic lab. The whole thing came crashing to this isn't from Google, this is genuine. I took this picture myself. And every time they need to order something, they just stick a Post-it note on the wall, which is until they ran out of Post-it notes, and in which case that ordering system shut down, and nobody had anything. It just became the Wild West. I always remember my supervisor, who had this big corner desk, and you would walk into his office, and there were just reams and piles of paper all over the place. It was like stepping into some kind of academic Narnia. And there were all these papers and things, and he'd have about 12 inches in the corner of the desk, which was for his laptop that he would work on, hunched over in one corner. And that was genuinely how we organised work. For this reason, in round about the second year of your PhD, somebody wanders into the lab, they put a sack over your head, and you're packed in the back of a van, and you're taken to a graduate school. And they sit you down and say, right, everything you've been taught so far is terrible, you have to write a Gantt chart. This person's never done Prince 2 project management, by the way, and never actually done a project, but they need to figure out how to submit your thesis, because PhDs are kind of strange. They're a bit like a pass-fail, and the only metric that universities have of whether you've done well is whether you submit on time. So the university has a vested interest in making sure that you get there on time, and this is their best guess on how to do it. So I sat down, I did my first Gantt. Sadly, it was a long time ago, and I don't have my first Gantt chart. But it's a similar thing to this. You kind of develop a method, you then do a load of science. Hopefully, at the end, you kind of graduate. However, I was sat there writing it, and all of the time I was thinking that everything I know about science tells me that this isn't going to work. But you do it anyway, because that's what you're told to do. But everything's telling me, I've been in the lab, I did my first year, I've done my second year slump, nothing's working. How can I accurately predict what's going to happen, right? There's a great book called The Personal MBA. If you're crap at business, which I am, it's a really good book. It's a bit like a cheat sheet of everything you need to know. I just love plugging that book because it's really useful. However, experiments can fail in 3 different ways. I'm skipping ahead of myself. I do that, I get overly excited. Experiments, really, when you're kind of doing research and it's brand new and you've no idea what's going to happen, there's kind of 3 things that can happen at the end of your result. The first one, the holy grail, is you have a successful success. And that is you planned that something would happen, and that thing actually happened. That happens about 1 in 10 times, and I'm not joking, that's probably optimistic. But that's generally how often it happens. Secondly, as NASA would put it, you have a successful failure. A successful failure is something happens that you didn't expect, but actually you learn something new out of it. And that's useful information that you can document and record and share. However, you also just have failure. kind of abstract failure. And that is the technique or the experiment, or for some reason you do not know, your experiment fails and you have no idea why. Often that isn't useful, and it isn't useful for 3 different reasons. This isn't a conclusive list, but there's a couple of reasons why that might be. The first one is what we call the golden hand principle. It's synthetic biology and working with microorganisms is still a practical skill. We still have to interact with it with our hands, which means you can get good at it. And if you've met scientists and their ability to use their hands, getting good at it can be a slow process. But actually, some people are good at it, some people are very good at it, some people are still learning, and I definitely put myself in that category, which means there's certain aspects that you perform when you're pipetting liquids and moving things around that mean you're more likely to get a success than not, because you just physically are better at performing the experiment. There's also technical complexity. The research that you're trying to perform is complex, and by its very nature, it means that you have to think a lot about things that can happen, which means you are prone to failure, because actually you could miss something. You could miss a certain variable that you haven't accounted for, and the thing can fail. And finally, there's whether you've performed the correct sacrifices. That is a common joke amongst labs. We don't actually perform blood sacrifices anymore, but what it kind of sums up is this concept that things will fail just because we don't know. And actually what scientists mean when they kind of joke and say, well, it's a bit of a dark art, you've just kind of got to figure it out, is everything is a variation around a mean, and biology is no exception. And we have this kind of normal distribution. A lot of you all know what I'm talking about here, I hope. But we have this normal distribution of everything that life does, and you're hoping that your experiment lies somewhere in the middle and that everything will go according to plan. But of course, there are outliers that we have to account for. And those outliers can cause an experiment to fail in a way that you don't expect. And that's just the problem with research in general, and probably stuff that you're familiar with. However, there are some very specific examples in microbiology that cause it to be hard to predict as well. The first one, again, talking about a normal distribution, every single life is different. If me and all of you drank a beer right now, it would affect us all in slightly different ways, right? We're all different builds, we're all agreed to different heights, we're all used to drinking in certain ways, which means we have different tolerances, etc. And we call this biological variation, which means that actually you always do multiples of your experiment, or what we call a biological replicate, which is represented by this green test tube here. Here I've grown 2 bacterium. They're both exactly the same species, but I'm growing them separately because I need to account for my biological variation. Biologists traditionally are very bad at maths, so we use the holy number 3 at all times. Everything is a triplicate. There are mathematical ways of figuring out how many replicates you need, but most people don't use them, and I won't go into it here. You then have technical replication. As I mentioned before, the thing you're performing is technically complex and relies on often instruments to be able to measure things. And those can be wrong to a certain degree of error. So then you have to kind of do replicates again on the technical aspect. So each one of these biological replicates needs to be measured several times. And then you can start to account for all of these problems. On top of that, you're working with living organisms, which means it takes time. This here is what we call a growth curve. And they're fairly universal, but the time element changes. And actually, whenever you start an experiment, you have an element of lag, and that is where the organism kind of gets used to its environment. It chills out a bit, figures out where it is in physical space, and it kind of decides whether it wants to grow or not. You then have what's called a logarithmic phase, where the organism figures out that it's got everything it needs, it's having a great time, let's divide and divide and divide. And a lot of these organisms divide through what's called binary fission, so they split in half. Not all of them, but a lot of them. Which means that it's logarithmic, because every one splits into 2, etc., 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 and the organism grows. You then encounter what's called stationary phase. That is where your nutrients become limiting, or perhaps they're just so packed together they don't like it anymore, and the amount that are dying match the amount that are growing, so your graph levels out and you start to hit stationary. Most of your experiments end here, but if you're doing something very particular and you just kept this going, eventually you enter what's called death phase. And death phase is where there's more dying than there are growing, and your numbers go down. Why I'm trying to drive this point home is if you're doing synthetic biology and you're cutting DNA together and you put it into organisms, you could do an hour and a half's work and then be stuck behind a 12-hour cycle of letting the thing grow. Right? So all of a sudden you're adding a huge time element to any experiment you're doing. And that will come important later on. There's also positivity bias, which this relatively complicated diagram is trying to tell you. But all it means is that in order to secure a good job, particularly in academia, you have to publish. And we push things out in papers. And we publish it. And the problem is it only kind of sells and people only read it if it's really good. Which means you publish all the stuff that works. And all the stuff that works get pushed out and everyone reads it and thinks, isn't this amazing? What you don't do is publish anything that fails. Under no circumstances must we communicate that anything has failed at any point, which means that I could be working on stuff right now in my lab that somebody's already done 10 years ago and it never worked. You just don't know, right? And one of the things I love about the developer community is everyone's got a kind of a blog post or something saying, we tried this and it was crap. There's less of this positivity bias, whereas in biology it's a real problem to getting work done, is that you don't understand that something's already failed before. And this hits you right in the Gantt chart. You've probably heard of this before, but just to go over the basics, this guy, or whoever's doing this, spent up to kind of a year and a half developing a method. And that would be some empirical work, but a lot of that would be planning, whiteboarding, that kind of stuff. You've got no idea that it's going to work, right? Because you haven't tried it in the lab, so you don't really know it's going to work. So that means all of this kind of experimental work they've got in their mutant screen, it's going to extend because you're going to have problems, you're going to have to run it again. Which means all of a sudden your kind of submitting of your papers gets pushed out and missed entirely. On top of that, you're writing papers, but how do you know that anyone's actually going to want to read it? There's no point that you're actually doing value delivery, right, in biology. You're just kind of assuming that it's interesting to people. And finally, you have all of this pushback from various things that have failed, and you graduate here, and if you miss that, well, it's game over. Right? This isn't going to be new to you guys. It's a concept that's very well versed in business. People like to use the term VUCA these days, things that are volatile, uncertain, complex, and ambiguous. And there's a cool kind of book called Team of Teams that I used to inspire kind of leadership stuff in our company. It's quite old now, but it dives into these concepts really well. So even at that early stage within academia, I started to get this impression that there must be a better way for dealing with complexity, and there must be a better way for dealing with biological experimentation. I started to come up with these mad flowcharts, and this is just an example, but these would be kind of an A3 page of all the outcomes that could possibly happen and trying to account for them, and actually to follow them with just color coding as to which direction it was going in. And it wasn't based on any prior literature, it's just the only way I knew how, so I was kind of grasping at anything I could. I eventually wrote my thesis, and at the end of your thesis, if you do well, they basically say, yeah, it's all right, there's some minor corrections you have to do. And that in itself is a bit of a project. So I experimented, having chatted with Singer during an internship, with applying kind of agile project management to my corrections. It was really easy because it was just correcting something I already knew. There was loads of known quantities. And it was only me involved in the project, right? So it's very simple. And this was the actual burn down chart, just brushed up in Excel, that kind of had all the estimated efforts through kind of an effort poker, which is really hard to do by yourself. And it kind of led this prediction. This blue line here is the prediction of what I would need to do to hit my deadline date. And actually, by applying this technique, I delivered early, which was exciting at the time, but I didn't know if it would actually be useful from a biological point of view. Again, this was just writing something. So that is a little bit of an introduction into biology, kind of the problems that are associated with biological projects and what they mean. And now I'm going to talk a little bit about how we tried to roll this out at Singer and some of the problems we've encountered and kind of what Singer do as well. Which is section 2, learning to walk before we can sprint. This, in a nutshell, is Singer Instruments. So at the top left there, we've got my old lab, which was kind of a little cupboard that we worked in. There's me there. This is Jose, who's our PhD student, who sadly is finishing this year. And we have a long string of interns. So roughly every 3 months, we have a PhD intern come in and work with us. And this is Heather and Lucas that came to join us. And this is Phil, who's our in-house scientist. And this is our MD, Harry, in our new lab. So we got kitted out in a new lab, so something we were doing must be working. And we got kitted out in this new lab that we extended into. And doing what we do and being kind of culture focused, we managed to win a couple of awards. This is me being kissed by our engineering manager. And these are a load of people who do amazing work that sadly I don't have time to go into, but they're all amazing. And ultimately, what Singer do is create robots to speed up the lives of microbiology. So we design, build, and test robotics that scientists can use in their lab to speed up their experimentation. And we are kind of the research group. We're a very small team of scientists. We average between 3 and 5, depending on what the interns are doing at the time. Which means we get up to all kinds of weird and wonderful cool stuff. So this top one was an intern called Robert Crawford, who at the time was at the University of Manchester. And he took a colony picking robot, which is picking individual yeast colonies that you can see up here, and placing them on agar plates. And we developed a new feature that meant that you could basically input a CSV file into the robot, and it would print anything that you kind of threw in there. And we thought, well, let's use this to create bioart. We've got loads of different manipulated strains that can fluoresce in different colors. So if you put different lighting modes on them, the organisms themselves actually glow. And if you want to get somebody's attention, make it glow, right? That's the thing. So he basically created a really simple script that took a bitmap image, stripped it out, put it into coordinates, and then fed that into the pixel, which meant we could start to print pictures using living organisms. And here you can see we've got a DNA structure that's been printed with that. This here is all the same Petri dish. This is a Petri dish here with all of our yeast colonies growing. Some of them are expressing something called M-cherry, which is a red fluorescence, and some of them are expressing what's called BFP, or blue fluorescent protein, which means if you swap the lighting modes in Pixel, what we call disco mode, you'll be able to get the Singer logo flash up in different colors, and you put it together as a GIF. It's a really cool thing that we work on. And it sounds like we were just goofing around, but actually what we were doing is creating this kind of knowledge that we could get the Pixel to do anything, as long as we had the right CSV file to begin with. And it wouldn't crash in the process, which is good. About roughly 10% now, used to be 20% of everything we do is academic, so we get some funding in that allows us to do academic work, and we put the robots through their paces by driving that. I'm going to talk a bit about this later on, but this is Jose's project. And what you're actually seeing here is an agar plate, which is like a jelly solidified plate. And we've got a whole host, there's actually a 1536 plate, a whole host of different organisms on that plate that are growing in brewing conditions. So I think this is actually wort media, so it's actually similar to what you'd have in beer, but solidified. And what we're seeing here is wild yeast strains grown on these plates over 240 hours, and we're showing that some of them grow better in that condition than not. We can take regular pictures of this and analyze how they're growing, which gives us an idea of whether a yeast strain is going to be good for brewing beer or not, basically. But we still had this problem. We still had to predict biology. People tell you that when you join industry, all the problems that you previously had of things failing would go away. That's simply not true. You're still working on the same organisms. You still have all the same problems. So all the problems we discussed earlier of predicting biology were still there. Our project management was slightly better. To begin with, we used a program called Liquid Planner, which is very Ganty, to be completely honest. But what it did is it afforded us empirical data. So we could log all of our experiments there, we could log how much time we were taking on them, and all of a sudden, we had a number. And that number could predict how long our experiments was going to take. So we figured out that we could have a test plan objective. So when we test a new robot, we build up a test plan. It's filled with objectives. There's roughly 9 to 14 of them. And we realized that a single one of those could take anywhere between 27 and 30 hours. A single experiment lasted somewhere between 6 and 8 hours, depending on various things, but these were kind of the approximations. And we thought, great, we could predict experiments. We had time. It was based on empirical data. As scientists, we like this kind of thing. But it wasn't without its problems. Firstly, definition of done. We kept delivering things that were not quite right. So we'd get to the end of our experiment and say, look at this cool thing we've developed. And people say, well, that's not quite what I wanted. Secondly, we had shifting requirements. The loop that it took, those 30 hours it took for us to get through an entire kind of objective, by the time it's done, priorities would change, and actually we'd be behind the curve all the time, and we were constantly playing catch-up. We also had shareholder expectations, so we'd sit down and say, cool, I've predicted your experiment, this is how long it's going to take, and people would say, well, 30 hours sounds like a long time. Well, yes, but that's how long it's going to take. It's confusing. And also, I was really struggling to empower my team, to be totally honest. Using this kind of Gantt-based system, I had to be in complete control all the time of everything that was going on and understand where everything kind of fit in, which meant my team were kind of just coming to me and saying, well, what am I doing next? What am I doing next? And that is a terrible way to deal with clever people. Because actually, PhD scientists, and I think a lot of the people in this room, they're creative and they're exciting and they're interesting, which means they work much better at a very loose kind of informal structure of being able to rely on other subject matter experts. And things get done much faster when they're free to do that. So I needed a way to kind of empower the team to be able to realize what was coming and do it for themselves. And I remember the conversation that I was chatting with Phil at the time, and I said, hey, why don't we just try Agile? Well, as far as we know, it's never been rolled out in biology before, but things can't get any worse. We may as well give it a go and see what happens. So we came up with some primary objectives, very scientific, that we'd know that things were working if we could do these things. And those were mainly empowerment. So we wanted to empower scientists to see what was coming up next in a created and prioritized backlog. We'd learned a bit of lingo by this point. And we wanted to choose our own work. Secondly, we wanted to communicate internally what was going on, what is going well and what isn't. Basically, that internal communication piece that we were missing because we were people were reliant on me knowing what was going to come next. Thirdly, we wanted to predict how long an experiment would take, and in particular, how long it would stretch on into the future. We had a lot of inputs from loads of different departments, which meant that we were kind of always saying things will be done next week, things will be done next week, and we were kind of struggling with that. And finally, we wanted to use analytics to produce a dashboard as a talking point, because we kind of wanted to, we had this idea that we'd push out monthly reports so we could keep stakeholders involved as they went along. So can we sprint? No, would be the first answer. This is the first sprint we ever did, and being scientists, I think it's called Initial Sprint Test. The name's got much more exciting from that point onwards. And at first we thought, hey, we can't do it. But the team were really excited by it, and we learned some really interesting things. And actually they preferred the way of working, even though it was a horrific failure. But we learned a few things. Firstly, we learned that we needed to granularize. We kind of would just come out with this huge piece task where we're like, hey, we're going to nail putting a piece of DNA into yeast and getting it to work. There were just far too many variables, and we were never able to kind of finish some of those huge pieces of work. Also, we realized that we really liked effort points, and this is from Jira, by the way, and it uses story points, but we really liked the idea of things being a relative measure. And that was because it turns out we were really crap at figuring out how long something was going to take. So we tried time estimations, and it just didn't work. So we actually really liked kind of that relative effort, because we know how hard something is, and we know how hard something else is, and we could kind of make an approximation based on that. Also, we had to be realistic. We were very, very optimistic with the amount of work we could get done, and that was probably the cause of a lot of our problems previously. And we realized very early on that we were never going to get to the end. This massive scope change here was actually one of the scientists was on holiday and took on a load of work when he came back. So that was less of a worry than it seems at first, but it became a worry later on for different reasons. Also, we were sprinting for 4 weeks, which may seem like an age for a lot of people in this room, but actually with all that kind of growth curve problems I was mentioning earlier, it kind of felt right. We'd had no other explanation other than that. It wasn't empirical, but we decided to stick with it. And other than that, everything we were doing wasn't so different from all the other kind of agile talks you'd have. We were meeting and we were doing kind of effort scoring, and we used effort poker because that was something we learned about on the internet. It worked fairly well. In fact, now the team's pretty instinctive, and we're pretty in sync with how hard we think something is. So we don't have to go through effort poker all the time. But if we have a new member of the team, or we have an intern come in, it really helps to go back to basics and kind of teach them kind of how we do effort scoring. That would all be fed into an experiment or thrown into a big old backlog. We'd spend 4 weeks chugging along on it. And at the end, we'd just have a retrospective of what went well, what didn't. We'd change it all up and move things around. More recently, we've come to a very targeted sprint kind of scrum team as well. We used to have a much bigger team with all server support and quality. But we've really found that communication on what we're currently working on is vital. So we do a lot of that as well. So not so different to all the stuff you're going to be used to. And we started getting results. This was only our second ever sprint, and I could show you millions of these, and I won't. But we started getting results. Some people are frowning at this. I love that. We had a few problems still, and that was that everything kind of got done last minute. There's 2 reasons for this, and we're not really sure what they are. Partly, some of it is what I mentioned before with that lag phase. Things are growing. So actually, we start an experiment here, and you're not going to get results till 2 weeks later. So you kind of have this thing that we need to think about how we granularize and how we do work. We still haven't figured it out, but it's something that we've noticed throughout all of our sprints. So that's part of the answer. The other part of the answer is that we were really struggling with our definition of done and scope. Because actually, what would happen is if you were doing an experiment and you really enjoyed it, you'd keep doing it. And you just keep working on it and keep working on it until you kind of thought like, oh, the end of the sprint's coming up, I best finish this. So people were holding on to work. So we really had to scope out the projects that we were doing far better because they were too open-ended. We were asked a very interesting question. I showed a couple of these slides to a group called the British Yeast Group, a science-based conference. And actually somebody must have been an Agile practitioner out there somewhere. They weren't just scientists. Because somebody asked us the question is, what does technical debt look like in biology? Which is a very good question that we are still figuring out. And we think we're starting to get inklings of what that might look like. And I'm hoping to share with you as we go through. So that was kind of section 2. We were learning to walk before we could sprint. And section 3, I'm going to go through an actual sprint. So the kind of stuff that we demo and the kind of stuff we go through. So an actual sprint. What does biology sprinting actually look like? So this is all demo slides. All the work was done by other people. I was just there at the time, so I'll introduce it as we go through. But Jose, who was doing his PhD, went to a local cider farm called Tor Cider. And he went around in a field and he sampled a load of trees, leaves, and the pressing room, which is over here somewhere, and loads of different areas. And we got all their geographic locations. And he took all of that data and he grew a load of yeast strains. So in the southwest, there's a really cool thing that people do called wild fermentation, which is where basically you throw a load of apple juice in a vat and you just leave it to ferment. Which scares the hell out of the brewing industry, but it's kind of how we do it, and it's how it's been done. So the cool thing about that is you get really interesting aromas and flavors, and we were funded by Horizon 2020 to do just that, to kind of figure out if there were new aromas and flavors of beers and wines and ciders that we could introduce. And he sequenced all of this data. So I mentioned DNA sequencing earlier. And we came up with this massive pie chart that had all of these different organisms and their relative abundances based on where they came in this field. And we could segment that data and find out where these strains were coming from. And what we're doing right now is following that through and seeing if they appear in that fermentation process, wild fermentation, if one of them dominates or if it doesn't. And then we performed something called the Sniff Test. We knew we needed to get data out within our sprint, and we needed to figure out very quickly whether the aromas were pleasing, whether they were good, whether they were bad, and kind of what we need to focus on. So we got loads of people lined up, and we gave them all 9 tubes. So 2 of the tubes were controls. We had 5 wild yeast strains in there, and 2 were secret control tubes who had some brewing strains in there alongside them. And we just gave everybody a basic format kind of questionnaire to fill out. All of the species are there, but I won't go through them. But we've realized there were 4 that were really interesting. All this work was done by Phil Kirk, who did all the computational methods in the background. But we had, for example, Saccharomyces Cerevisiae, which is actually used in brewing fairly commonly. But when people took a small tube of this and smelt kind of apple juice with this, they were reporting that it smelled woody, some swear words, they smelled bread, pukey, kind of hungover, the smell of feet, this kind of stuff. So generally not the kind of thing you'd want to drink. Generally, people said it smelled worse than kind of the control tubes. And often they said it smelled rotten or vomit-like, which sadly is a fairly common occurrence for a microbiologist. We then had something called Pichia cluverii, and overwhelmingly we had this response of pear. It smelled like pear. Pear's pretty good. Also rancid, but we'll go with pear. And actually, when asked if it smelled better than controls, the party were divisive. So it actually split between worse and better. Divisive is sometimes good. It's a conversation starter. So we thought that's interesting for a wild strain. We'll take that forward and see how it performs at a later date. We had Hansenaspora uvarum, and overwhelmingly we had acetone. There was some cider, chemical-like restrictions, but we had this acetone smell. And generally, people said it smelled more chemical or solvent-like. This is really interesting from a biotechnological point of view because we can take that forward for further study, find out what's in it, and see if we can make a link between what people are smelling and what it's actually producing. So it's quite interesting for us from that point of view. And finally, Saccharomyces Cerevisiae. This is brewer's yeast or baker's yeast, but it would be a different strain because this one was wild. But it's kind of the thing you normally use in kind of your ale fermentation, if you like. And generally, people thought it smelled better than the control tubes, and also that it smelled sweeter. So we took that forward for further study as well. And in order to try and encounter a problem, brewing strains have spent hundreds, if not thousands is a bit of a stretch, but definitely hundreds of years in brewing conditions getting used to them. We've almost tailored brewing conditions to them, and they've grown up and evolved to be in that environment or been selected in that environment. We didn't have that time, so we did something called random mutagenesis. which is where we put them all in brewing conditions, and then we threw them in a machine called the rotor, and we actually used a UV bulb, which is used for cleaning the rotor, to induce mutation. I won't go into how that works, but it causes the organisms to mutate. And then we selected survivors and basically selected them in higher levels of ethanol, which is just kind of alcohol you get in your beer to kind of grow. The idea being is if we would artificially selecting for those that could survive at higher ethanol levels, which meant that they would be more likely to be a brewing strain. And we were pushing that. So we were physically changing the makeup of these organisms to make them better. And we got good results. So within 10 minutes of UV, we noticed that we had survivors all the way up to kind of 12 to 16%, so kind of like your wine strength kind of things, if you like. And while this worked and we got this is where our technical debt came in. So we could produce these strains, and these strains do exist, but we tried exactly the same experiment again, and we didn't get the same results. So while we'd done it once, we couldn't do it again. And all of a sudden, our repeatability became the problem. So it wasn't so much the documentation and making sure that that was there. It was whether we could repeat the experiment in a robust manner was kind of where our technical debt started creeping in. So we had to start thinking about matrices of experiments, and could we actually perform experiments in multiple different conditions at the same time in order to kind of account for that. So that was an actual sprint. That was 4 weeks' work that we kind of pushed out to a conference a little while ago. There's a very short section as I come to the end, which is getting back in the garage, achieving our objectives, learning something from the past, and some foreshadowing as well. So we had a velocity chart. It was a thing that existed, which meant that we could sort of start predicting how long these experiments were going to go for. And as long as we were doing our ethic planning, we could predict into the future of when we'd get that next paper out, or when we'd be able to develop a new feature. You'll notice that one of the names is redacted. That's because it will become important later, but that's a secret project the R&D team's working on. It's no swearing involved, I promise. But generally, it meant that we could start to predict. We actually had new members of the team joining, so occasionally things shot up. But generally speaking, we knew what we could kind of get through and whereabouts we'd end up if we effort projected enough into the next quarter, which was exciting. Did we hit any of these objectives? Well, yes. We were empowering scientists. People were deciding what work they could do. And if nothing else, that was a success story that we could take home. Were we communicating what was going well and what wasn't? Yes, because we were doing retrospectives and we were having our daily meetings. And all of a sudden, there was a conversation around what was going well and what wasn't. And if nothing else, we were achieving that. We could start to predict how long these experiments were going to take and start to predict into the future. So if somebody comes to us and says, look, this is really important, you have to do this experiment right now, how long it's going to take you, we had a basis at which we could talk about that. And it wouldn't just be, it's going to take you 30 hours, see you later. We could actually start to talk about how hard this thing was relative to other things as well. However, were we communicating and pushing out communications? One of those names earlier were redacted, and that's because it was a secret R&D project. So we were the research team, but actually there's a huge engineering team and a huge software team that dwarfs our teams entirely. And they were working on something really exciting and really interesting, which we will call the mystery box. And Elliot, who's represented here by Mr. Burns, who's our R&D manager, came to me and said, look, we're working on the mystery box project. We want to get involved, and we're rolling out Agile. They brought in an Agile project management leader. They rolled it out very quickly. They were working in sprints, and they were doing really exciting work. And we were confronted with a quite interesting question. We'd built a lovely system. It worked. We were predicting biology. Everything about it was exactly what we wanted and what we'd envisioned. But was it ritualistic? Yes, we were following a pattern that we then kind of could iterate on and move on. But it was ours, and we kind of liked that. But in a way, it made us siloed, because actually our work was contained, and we discussed it amongst ourselves as a group, and perhaps what we weren't doing was pushing things out to the wider business. If we really wanted to deliver value to the end user, we needed to work with the company more widely. So actually, did we need to break apart our lovely system that we've become accustomed to, forget about the ritual, and go back to actually the philosophy of Agile and actually what it means and what we're really trying to do by almost disconnecting our kind of siloed mentality of this is how our work structure looks like and bring it into, well, how do we deliver this thing that's going to be meaningful and useful? And I'm not going to answer that question because that's for you to think about and hopefully talk to me about later on. But we're still deciding, and we still have aspects of this. And I think the true answer, as always, for these complex problems lies somewhere in the middle. So that was section 4, kind of getting back into the garage and learning from that startup mentality. And with that, I just want to say thank you very much for taking the time to listen to me. I know it's a bit of a rant, and you're probably used to a lot of this stuff already, but we found it really exciting. So hopefully you found it exciting too. I've got to thank a few people. Firstly, Jose and Phil for doing all the work, kind of our academic collaborators in Daniela at the University of Manchester, and all of our interns who've done amazing work, and the cider farms that work with us, and of course the European Commission for paying us to do the work in the first place. So thank you very much.
Question or two? Maybe one question? Anybody got a question?
If anyone's got a question, I'm very happy to take it.
If not, no question.
Go ahead.
I spoke with someone from BioNTech, and I think they're doing similar stuff.
Yeah.
What they're doing a lot is using lean techniques like 3D printing.
Yes.
To speed up some of the prototyping and stuff.
Yeah.
Have you guys done something similar?
Absolutely. So in research specifically in our group, we have Frank. Frank is our 3D printer. Is the hardest working member of our group. And yeah, we 3D print little tiny kind of contraptions all the time in loads of different experiments. It's really useful. The engineering team have a whole bank of them. They're Pursa, can't pronounce it properly, Pursa 3D printers, huge bank of them that print all the time. And iterating hardware is a whole talk in of itself. But yeah, but very interesting.
Very cool.
Yeah. Go ahead.
What made you turn to Agile?
It's a really good question. I had done an internship with Singer before, and I was quite close with some of their software team. And while they weren't working in Agile at the time, a lot of the practices had started to bleed in, right? They'd started to learn about it and kind of talk about it, which meant I was in a lovely kind of environment where people were in a kind of safe way, kind of experimenting and kind of trialing it. So we'd realized that the alternative didn't work. We'd kind of done a bit of Prince2 and kind of thought, this is really good for building sewage systems, but not so good for biology. So we tried that. We just tried it, and it seemed to work. The sales and marketing team were also rolling out something very similar, so we could swap ideas. And I think that cross-pollination, if you like, of different ideas was just too good an opportunity to mess up.
Okay, so let's have another big round of applause, please.
Thank you.